Hello. Today I'm going to review genetic notations and Mendelian genetics for blood typing. So remember, when we look at blood typing, we've got the ABO type, and then we've got the positive negative type. So what we're looking at here are two separate genes. Each of them are coding for a, each of them are coding for a an antigen to be on the surface of red blood cells. All right. Now, when we're looking at the ABO type, we've got two dominant genes and one recessive. When we're looking at the Rh factor, or it's also given the notation of D, the letter D, and we're talking about positive or negative. That's it. All right. This all harkens back to when you learned about Mendelian genetics and the cross types and things like that. Remember doing these in the Punnett squares in biology. So I'm going to review some of these things here. You got a couple of concepts here, dihybrid cross for one thing, and you're also looking at codominance when we look at the ABO plus minus blood types. All right. So the letter I is used. Remember, it's capital, means that we're referencing the dominant gene. All right. Lowercase here means we're referencing the recessive gene. Now, to have it is to have an antigen, okay, that's ABO type, is dominant. There are two types of that dominant gene. One encodes for type A and one encodes for type B. All right. So there's two different types of dominant gene here. And they mix together. If they mix together, both of them are expressed. So you get the A antigen and the B antigen. So the antigen is just a protein that appears on the surface of red blood cells. Um, that's it. Okay. So let's take a look here. And then the positive, negative. When we look at people's blood, the A negative, B positive, and whatnot, we're looking at another gene. This is another gene altogether with two different versions or alleles, positive or negative. Okay. So the capital D is referring to positive, that's dominant. Small d is referring to negative, that's recessive. All right. It's called, the D is also called the Rh factor. Rh stands for rhesus monkeys. Rhesus monkey blood used to be used to figure out the plus minus, the negative, positive of people's bloods. So that's how it got that name, Rh factor. Rh, rhesus, Rhesus is rhesus. So, but we use the notation D because remember, when we do Mendelian genetics, it's only one letter that we use, right? So when we talk about somebody who's type A positive, right, we're looking at type A positive blood, then we've got four different combinations, possible combinations. So the A positive, we say that person is type A positive, we're talking about their phenotype. Remember, the phenotype is what you see, what you feel phenotype. Genotype is the genes. What is the gene combination? What do they have on their chromosomes? What gene, what allele do they have for that gene do they have? All right, so somebody who's A positive. Remember, we put it down twice because one chromosome from mother and one chromosome from father. Okay, same thing. So it could be that they're both homozygous. Homo meaning the same, zygous meaning zygote. The allele for that gene is homo. They're the same in the zygote. All right, when the zygote gets formed. So they can both be homozygous. One of them can be heterozygous and one homozygous. Okay, so like for A, it's homo. For positive, negative, it's heterozygous. Remember that when both genes are present, dominant and recessive, the phenotype is positive, is dominant, so it's positive here. Recessive, we don't know what the other one is. We can't be sure of it. So they can be heterozygous for A and homozygous for D, or they can be heterozygous for both. So these are the four possible combinations of somebody who's A positive. All right. So for example, somebody who's AB positive, A and B, when we see AB, we know what the two genes are. We know what the alleles for those genes, for that gene is, okay? On one chromosome it's for A and the other chromosome it's B. We know that has to be the case. They're positive, could be two capital D's or a big D, little d, okay? Could be either one of those. Could be heterozygous or homozygous for the positive factor. So somebody who's AB positive has these two combinations. They could have those two combinations of blood. Somebody who's AB negative can only be little d and can only be AB. Just like somebody who's O, so somebody who's type O, they are both recessive for the ABO antigen. They're O, they don't have any antigen that's A or B. They don't have either one. And then they don't have the D factor, RH factor either. So they don't have any of those antigens on their blood cells, right? So if they're O negative, we know that's their genotype, and that can only be their genotype, right? Okay. So let's take a look at that first problem. Looking at this first problem here, Susie, blood type AB negative, had a baby with blood A positive. Could Bert be the father who's type B positive? Okay, type B positive. Let me see if I can just shift this over a little bit. Anyway, that's good enough. So let's take a look at this. Now, Susie, we just said this a minute ago. If she's AB negative, right, she's only going to be that. That's all she can be. Okay, we know what she is, genetically speaking. Bert is going to be like the A positive. There's going to be four combinations here that's possible for Bert. Okay, well, let's start off with both homozygous. D, because he's B positive, okay? Could be this. Could be this. And could be this. Make that a little bit. Oh. Okay. So those are the four combinations that Bert could be, okay? Now, taking into account all of these, which is the most diverse? You can tell the bottom one is the most diverse because they're both heterozygous. So he could have any combination in his gametes of that we have here, okay? So let's take a look at a Punnett square with this. Let's draw the Punnett square. We'll just keep that one. That's one possibility for Susie, right? The other possibility is IAD. So now we're looking at the gametes. Remember the gametes? So for Bert, there's four combinations of gametes. He could have one for dominant, okay, and then both dominant, one dominant, that one recessive. Oops, looks like a Z here. Oops, done with the dominance there. So then we could have recessive, dominant, recessive, recessive. So we can basically have dominant, 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 recessive, recessive, dominant, and recessive, recessive. Okay, those are the possible combinations. No matter what of these he is, okay, you can't get anything different from these. So we don't know what he is. So the most or genetically we don't know what he is specifically. Okay, so we have to go with all the possibilities of gametes he could make, and there's the possibilities. That number would go, those numbers, the number that there are, would be less if he was any of the other, but the lower one is the most diverse, and where he's heterozygous for both. All right, so those are the four combinations that Bird could have. So let's start pairing them up. So we got IB, IB, big D, little d. That's somebody who's B positive. Okay. IA, IB, big D, little b, a little d. AB positive. Somebody who's AB positive. IB, IB, little d. B negative. IB, IA, um, little d. So that's somebody who's AB negative. Okay. Moving on, we got IB, little I, big D, little d. That's somebody who's B positive. Here we've got A, little I, big D, little d. Okay. That is type A positive. What type were we looking for the baby? Could the two of them have a baby who was A positive? There it is right there. So yes, they could both have, together they could have a baby who's A positive. All right. I'll just finish this out so you can see the other combinations. This would be B negative, okay, and this would be A negative, right? But anyway, so could they have a child who's A positive? Yes, right there's your A positive, okay? So hopefully this is explaining it. Remember the notation for Mendelian genetics. Capital letter means dominant. Lowercase letter means recessive, right? That superscript there of A or B means the allele, the type of allele that it is. And you're putting them together in what we call a dihybrid cross. So when you're looking at the gametes, out of the combinations of the I and the D, you only get one of those two I's and only one of those two D's. Okay, but how they come together can depend on how many different I's there are and how many different D's there are. All right, so hopefully this will help you out with the Mendelian genetics. Just apply all those same principles to all those problems and should be good. Bye.